Okay, so now we're going to talk about additional forms of inheritance. So what we just went through was the most standard, like just dominant recessive inheritance, and how to create a Punnett square in order to express that. Now when we talk about incomplete dominance is when the dominant allele fails to completely mask the effects of the recessive allele. And one of the best examples of incomplete dominance is this pink flower here. Let's say that with this type of flower, red flowers is dominant. So white flowers is recessive. But if you have a homozygous dominant mate a homozygous recessive, and you produce heterozygotes, the heterozygote is a mixture of the dominant and recessive phenotypes. So the heterozygote ends up pink. So an example, if you mate two pink flowered plants, what is the probability of offspring being white flowered? So let's do a Punnett square here. Now it says that we're mating two pink flowered. So in order to be pink, we have to be heterozygote. And when we carry these in the boxes, this is what we end up with. We want to know the probability of having white flowers. Now white is recessive, so this is the only one that would be recessive, or that would express the white trait. So we have a 25% probability that they're going to be white. We have a 25% probability of red flowers, because if you look at these up here, these are red. And then you have a 50% probability of pink flowers. And in lab, we'll be going through many more examples of incomplete dominance. Another type of inheritance is codominance. In codominance, two dominant traits are expressed. And one of the best examples of codominance is our blood type. So let's think about the different types of blood types that people can have. We can have blood type A, B, AB, and O. If we have blood type A, this means that our red blood cells, and we'll draw red blood cells for all of these, our red blood cells have the A antigen on their surface. If we are blood type B, that means that our red blood cells have the B antigen on their surface. If we are blood type AB, this means that they have both A and B antigens on their surface. So, blood type AB is our codominant blood type, because it expresses both the A and the B antigens. And if we have blood type O, we don't have those types of antigens on the surface of our cell. So talking about inheritance of blood types, in order to inherit blood types, we usually put a letter I so we have the I, and then we have an A, and an I, and an A, or we have I, A, and little I. If we're going to inherit blood type B, it's similar. IB, IB, or IBI. Blood type AB is inherited having codominant traits expressed, IA, IB. And blood type O is the lack of an antigen on its surface. So now that we know this, and this used to be how we determined paternity, is just by looking at blood type. So if we want to mate an individual with type O blood with an individual that has type AB blood, what is the probability of them having AB children? So just looking at this and thinking about this logically, you're probably like, oh, well, if one of the parents is type AB, then sure, the children could be AB. Well, let's actually do the Punnett square here. If we draw the alleles or write the alleles for the type O blood, little I, little I. If we draw the alleles for the type AB blood, we have IA, IB. And if we draw our Punnett square, we have little I, IA, IB. And we carry these in. IAI, IAI, IBI, IBI. Now looking at this, 50% of our children would have type A blood, or there is a 50% probability of giving birth to a child that has type A blood, and the other 50% would be type B blood. So we'll do more examples of codominance once we get through the lab activities. Sex-linked inheritance. So when we talk about sex-linked inheritance, usually we're talking about inheritance of genes on one of the sex chromosomes, and predominantly that's going to be inherited on the X chromosome. Later on in this lecture, I'm going to show you some pictures of karyotypes, and that's actual diagrams and pictures of your real chromosomes that have been condensed and kind of blown up so that we can visually see them. And you'll see how tiny that Y chromosome is. The Y chromosome is so small that it doesn't really carry a lot of genes. So when we are talking about sex-linked inheritance, usually we're talking about inheritance on the X chromosome. If it is a Y-linked gene or trait, then only men would be affected because only men have a Y chromosome. So, sex-linked traits can be X-linked, carried on the X chromosome, or they can be Y-linked, carried on the Y chromosome. And if they are Y-linked, only men are affected. Because again, the definition or biological definition of a woman is, or of a man, sorry, is the presence of the Y chromosome. So if they have a Y chromosome, they're going to grow a penis, they're going to be able to produce sperm to serve in that role in producing offspring. So only men are affected if it's Y-linked. Now with X-linked inheritance, these traits are much more common in men. If we look at the sex, so for a female, we have X, X. And then we would put kind of a superscript to determine the gene. So XX with the little b, or a superscript uppercase B up at the top here, indicates an unaffected female. But if we have XB, XB, still unaffected. The only way to get an affected female is to inherit two bad X's, is what I call them. So two X's that are carrying the bad traits. But with men, you can have XBY unaffected, or XbY affected. So because there's two ways to produce an unaffected female, but just getting one bad B makes you an affected male, it's more likely, or the chances of being affected by these genes is higher if you're male. So let's do an example of sex-linked inheritance. So let's say color blindness. So you mate a colorblind male with a carrier female. What is the probability of having colorblind children? Let's look at the alleles here. So a male is going to be XY. Colorblind means that they have the recessive allele. A carrier female, well, female is going to be XX, and since they're a carrier, one's going to be normal and one's going to be lowercase. So when we do our Punnett square here, we have X B Y, X big B, X little b. And now we're going to carry these in. So here we go. Of these children, 50% are female, 50% are male. And when looking at this, of the females, 50% of the females are affected and 50% of the males are affected. Overall, looking at all of their children, 50% of their children are affected. In other words, there's a 50% chance that their children are going to be colorblind. If we look at this picture here, this calico cat describes a combination of sex-linked and codominance. So let's talk about calico cats for a second. So if we have a black female cat, we have XB, XB. If we have an orange female cat, we have XO, XO. If we have a calico female, we have XB, XO. This is the codominant type. And it's also sex-linked because it's carried on the X chromosome. Now if we look at males, we can have a black male, XBY, and we can have an orange male, XOY, but we can't have a calico male. Because in order to have calico, you need to have two X's. one X would carry the big B, the other X would carry the big O. Now because males only have one X, they can't be calico. Now there is a probability, or a very, very, very small chance, that we do have a calico male cat. Calico male cats will be infertile, and the way that we have calico male cats is through XXY Klinefelter's disease. In Klinefelter's, we have non-disjunction in the sex chromosomes, which leads to trisomy in the sex chromosomes. Trisomy means that we end up with 3 sex chromosomes instead of the normal 2. Now this is extremely rare, but since cats reproduce so much more than people do, and there's so many more cats than there are people really, we have this occurring about 1% of the time. So if you look at the population of calico cats, about 1% of them are male, but those male calico cats are sterile because they have Klinefelter's disease and they have that XXY. So now we're going to look at dihybrid crosses. In a dihybrid cross, you follow or track the inheritance of multiple genes at once. Now multiple, we can have polyhybrid crosses. In a dihybrid, di really means 2 genes, so we're going to stick with 2 genes for this, but know that these can get much, much larger if we're tracking the inheritance of multiple genes at once. So here we go. Let's say we want to track fur color and eye color. Now with fur color, black fur is dominant. White fur is recessive. And this is true dominance, so they either have black or white fur. So with dominant, they're either going to have big B, or big B, little b. While with recessive, it's going to be little b, little b. With eye color, red eyes is dominant. White is recessive. So in order to express the dominant trait, they would have big R, or big R, little r, and to express the recessive trait, little r, little r. Now let's say we mate. Let me put the example on the next page. So we mate a homozygous black-furred white-eyed individual with a white-furred heterozygous red-eyed, or we'll say a red-eyed carrier. What is the probability of the offspring with black fur and red eyes? So just like all of our other examples, we want to look at the parents and figure out the genotype of the parents. Homozygous black, white-eyed. with a white furred red-eyed carrier. So we have big B, little r, times little b, big R, little r. Now we have to randomly assort these genes. So we have to figure out the different combinations that could possibly be produced in the gametes of these individuals. So looking at this one, we would put this one with this one, big R, little r. We'd put this one with this one, big R, little r. Then we would put this one with this one, big R, little r. And we would put this one with this one, big R, little r. So those are our 4 possibilities. Now with these, we would put little r with big R, or little b with big R, little b with big R, little b with little r, and this little b with that little r. Now when we set up our Punnett square, these are the 4 possibilities on the one side, and our Punnett square has to be much larger. So going back here, we're going to put these 4 possibilities on the left side. And we're going to put these 4 possibilities on the top. And now we're going to carry all of these into the table. So we have big B with little b, big R, little r. And for this one, similar. Now if you don't know where these are coming from, we're just carrying in the big B, carrying down the little b, carrying over the little r, carrying down the big R. Now continuing that over here, big B, little r, little r. Now our question was, what is the probability of offspring with black fur and red eyes? Now we have to look at all of our offspring. If you look at all of our offspring, all of them have black fur. So that makes it a little bit easier. We can just focus on the ones that have red eyes. These here have red eyes. So if we look at these and we kind of circle them, these have black fur, red eyes, so 50% probability. And we will be doing another example of dihybrid crosses once we get to the lab period.